Hello, everybody, and welcome to the ESDR Kitchen. I'm Sprecher from the Department of Dermatology at the Tel Aviv Medical Center in Israel. And on behalf of the ESDR board and my co-chair, Professor Curdin Conrad, I'd like to very much welcome you to this new episode of the Molecular Cuisine of the ESDR Kitchen, where leaders in our field share their personal journey through scientific discovery. And today we are particularly delighted to have with us an outstanding world-renowned speaker, Professor Matthias Schmuth. Matthias Schmuth is Professor of Dermatology with a secondary appointment in pathology at the Medical University of Innsbruck. He received his MD from the University of Vienna, completed his internship at the Benjamin Franklin Medical Center, Free University of Berlin, and continued training in dermatology and dermatopathology at the University of Innsbruck. He was a research fellow and visiting professor at New York University, NYU, and UCSF, the University of California, San Francisco. Dr. Schmuth's research interest has focused on the morphology and molecular biology of epidermal differentiation and genodermatosis. His research team studies skin barrier function in disorders of cornification, and his laboratory aims to better understand the interplay between epidermal differentiation, barrier function, and inflammation, with emphasis on novel therapeutic approaches. Now, just before starting, I'd like just to emphasize that this episode is sponsored by Almirall. And before handing it over to you, Matthias, I'd like to remind everybody that we want this seminar to be as interactive as possible. So please forward your queries to the chat or the Q&A functions. And we will be happy at the end of Dr. Schmuth's talk to bring up those questions and ask Matthias to answer them. So Matthias, on behalf of all of us, thank you so much for the opportunity to hear you today. The floor is yours, please.
So thank you so much for the invitation to the ESDR kitchen. I'm just trying to share my screen. So the ESDR kitchen, it's around lunchtime. A gourmet kitchen offers several courses, even at lunchtime. Therefore, I'm going to talk about three different courses here. The appetizer is about how to customize topical lipid supplementation, sort of a lipid soup to begin with. The main course about developing gene editing, and a third course about how to target transcription factors for treating skin barrier disease. I've chosen these topics because the development of treatments for the barrier is lagging a little behind, those for inflammatory skin diseases, for example. So I thought it could be interesting to highlight the link between the bench and the bedside in this area. These are my disclosures. I do not get any of these funds myself. The most abundant cells in the epidermis, shown here, are keratinocytes. They have to specialize for what the task of the barrier is, meaning to differentiate in order to help protect our bodies. I don't need to go into too much detail to tell this audience that they undergo a program of dividing, migrating outwards, and then differentiating into corneocytes that form a structure important for skin barrier function, the stratum corneum, while at the same time retaining flexibility and allowing for various sensory and psychosocial functions. I like to call skin barrier function the final end product of this migratory effort that the keratinocytes go through during epidermal differentiation, which is a calcium-dependent process. This picture was taken in 1993. As a medical student in Vienna, I got fascinated with research and I was inspired to join a lab. After taking all my exams, but before graduation, I joined Peter Elias's team at the University of California, San Francisco, UCSF, to learn to study the skin barrier. I was so kindly guided in this new experience, supported while facing the many challenges in the lab, and while also discovering a new life outside of Europe. At the same time, I was introduced into the wonderful community of scientists in investigative dermatology. I was not only taught science, but the team in San Francisco also took me on hiking trips in the mountains, to concerts, and generally enriched my life. From Peter Elias, I learned about the shifting paradigms of investigating the skin barrier, first seen as an inert basket weave structure or saran wrap-like covering, then the famous two-compartment model, followed by more dynamic models of regulation and repair with strong links to cutaneous immunity, to the microbiome, and finally, the ability of the barrier to serve as a biosensor. The most recent example for this is the description of epidermal injury triggering IL-24 STAT3 signaling. This is Ken Feingold, an endocrinologist at UCSF. One important component for barrier function is the extracellular lipid compartment. And when I was at UCSF, I was assigned to a project with him who was studying fatty acid transport in other organs. Here you see the expression of fatty acid transport proteins, also termed solute carrier family 27 in cultured keratinocytes. There is selective expression of FATP4 in both undifferentiated and differentiated cultured keratinocytes, but not much expression of other fatty acid transport isoforms, perhaps a little bit of FATP5 in differentiated keratinocytes. FATP4 is also seen on Western blot of skin tissue, and the localization of FATP4 in skin in sections using immunofluorescence staining, you can see is expressed in the epidermis, in the sebaceous glands, and in the adipose tissue. The FATPs are one of several fatty acid transport proteins in keratinocytes that also include CD36 and FABP. Whereas knockout animals of CD36 and FABP5 are viable, there is decreased viability in FATP4 knockout mice, and they display a scaling phenotype and very tight skin. And this has received several different names by the different groups generating these mice: restrictive dermopathy, wrinkle-free, pigskin. 2009 then, FATP4 mutations in humans causing an ichthyosis phenotype were reported. As an example for the disease phenotype, I'm showing you this newborn that was presented to us in Innsbruck in March 2016. The mother had polyhydramnion at birth. The child did not do well with a low APGAR score and respiratory distress and hyperkeratosis of the skin, all of which actually improved over time. This was found to be caused by an FATP4 mutation causing this syndrome, also termed ichthyosis prematurity syndrome. So what do we know about FATP4? It is a membrane protein that is highly expressed in small intestine, esophagus, and skin. It acts downstream of CD36, upstream of FABP5. It mediates the import of long-chain fatty acids, and it's also involved in the synthesis of long-chain fatty acids. In the tissue context, we have shown that FATP4 is predominant in the epidermis. Keratinocytes preferentially transport linoleic acid over oleic acid. And the epidermis contains very unique long-chain fatty acids, which is shown here using mass spectrometry in a skin layer specific way. Here, the long carbon chains being linked to a ceramide backbone. Now, what is relevant for transferring our knowledge to the bedside is that on electron microscopy, we can actually visualize how the long-chain fatty acids are not attached to the circumference of the cells in mutants. The dark layer in the upper panel, marked with black arrows, is the so-called cornified envelope, and on top, we would expect a thin layer of omega hydroxylipid, which is lacking with FATP4 deficiency. In the lower panel, you can see that restoration of fatty acid transport can reconstitute this layer. Based on this, the therapeutic approach is to directly provide the missing lipids to the outside of the cells. This is an example of a specific replacement therapy based on understanding the molecular defect. The challenge is to customize these specialized lipids for topical use. After talking about FATP4 and lipid therapeutic opportunities, let's move to the corneocytes. Let's go to a different disease and a different pathogenesis, which affects the corneocytes themselves. Here is an electron microscopy image of keratopathic ichthyosis. This is due to a mutation in keratin 10. You can nicely see the cornified envelope as the dark, thick layer at the periphery of the cell. And in the lower right picture, you see the adjacent thin layer of long-chain fatty acids marked by arrowheads. So you can actually show this to be present. This is exactly what was lacking with the FATP4 deficiency before. Here, it is completely normal. So supplying the specialized lipids, the long-chain fatty acids, will likely not help in this condition. And in fact, in contrast to FATP4 deficiency in keratinopathic ichthyosis and in palmoplantar keratoderma, due to keratin mutations, the use of topical lipids is of little help. We therefore set out to try to correct the consequences of keratin mutations on the genetic level. In this case, keratin 9 mutations causing epidermolytic palmoplantar keratoderma. Keratin 9 is preferentially localized to the palms and soles, and when mutated, causes severe thickening of these areas, which impairs your ability to use hands and feet. As opposed to the normal keratin cytoskeleton network in normal keratinocytes, cells from skin biopsies from affected patients show keratin clumping, shown here in green. We adapted CRISPR-Cas to silence the mutation selectively on one allele, whereas the wild-type allele remains unchanged and produces normal keratin 9. This was based on prior work in mouse models, where siRNA had been shown to work by Leslie Pedroli and Irvin McLean, as well as Xiang Zhang, but that was only temporary, whereas Xiang Zhang also showed CRISPR-Cas to work in mice with longer lasting effects. So we tried this in cultured keratinocytes derived from palmar biopsies from healthy controls and affected patients. We nucleoaffected ribonucleoproteins complexed with the mutation-specific guide RNA for exon 1 of the keratin 9 gene into the keratinocytes and stained for keratin 9. As opposed to the normal keratin network of controls on the left, mutant keratinocytes show keratin clumping when you expose the cultures to heat as a trigger for the clumping, as shown in the middle. Thomas Trafoyer very nicely was able to introduce a stop codon selectively by nucleoaffection on the mutation-bearing allele, and as you see on the right side, the clumping disappeared. The keratins look like the wild type on the left. To bring this to the bedside, you have to watch safety very carefully. Besides the wanted nucleotide changes at the mutation, the enzyme, the nucleases, may be doing unwanted things. This is called off-target effect. We checked for this with Sanger sequencing and also with Cas sequencing, which is shown here. And we found that if you use the double nicking version of CRISPR-Cas, there are minimal such unwanted effects. To better understand the disease, we looked how RNA expression changes in the context of keratin 9 mutations. You see the cell types here. At the bottom, fibroblasts, in the center, 5 types of keratinocyte clusters, to the right, T cells, dendritic cells, and macrophage clusters. And circled in red, I point you at 2 keratinocyte clusters that look very different between left and right, between disease with keratin mutation and normal palmar skin biopsy. Not surprisingly, we found compensatory expression of other keratins, for example, keratin 5, 14, but also keratin 6 and 16 in the keratinocyte clusters. Interestingly, CalmL5, encoding a calcium-binding protein, was downregulated both in keratinocytes and fibroblasts of keratin 9 mutant palmar skin biopsies. Here you see confirmatory qPCR for CalmL5 in cultured keratinocytes, which shows a marked decrease in CalmL5, both in undifferentiated and differentiated cultured keratinocytes. Again, these cultures are derived from patient biopsies, and also the control biopsies were derived from patient biopsies from palmar skin. And we found the same decrease in patient-derived 3D organotypic cultures. So if you take the keratinocytes, culture them, and then make 3D organotypic cultures and look at the protein level, there was decreased CALM-L5 in these cultures. Interestingly, in the palmoplantar skin of these patients, we found CCL2 and CXCL12 increased in keratin 9 mutant keratoderma, both in keratinocyte clusters, but also in fibroblast clusters, as compared to a normal control. CCL19 and CXCL1 decreased in fibroblasts, whereas CCR7 was decreased in keratin 9 mutant dendritic cells. These changes in chemokines indicate, in addition to altered differentiation, that there is chemokine dysregulation. If you carefully look in the periphery of the hyperkeratosis in the patients, there is erythema in these patients, perhaps correlating with changes in these mediators. What does this mean? It is known that experimental disruption of skin barrier function results in cytokine chemokine alterations and dendritic cell activation. We and others have published this. Disease with chronic barrier impairment tends to be inflammatory, and conversely, you know this from atopic dermatitis, inflammation can also impair the differentiation of keratinocytes. For example, you see decreased filaggrin in inflamed atopic skin. As you are aware, this was described also in the genetic disorders of conification in the ichthyosis. Sometimes there's scaling without any sign of inflammation, as in congenital ichthyosis due to transcontaminase 1 mutations, but still you can detect subclinical inflammation in the skin biopsies of these patients. Sometimes you have overt inflammation, as seen in erythroderma with ALOX12B mutations. So in a skin barrier disease, such as the ichthyosis, most of the time, the skin also has detectable inflammation, and cytokines, including interleukin-17, interleukin-413, interleukin-22, have been reported by Amy Peller and others, making these potential therapeutic targets. And treating ichthyosis with biologics, such as antibodies against interleukin-17 and interleukin-413 and others, has been shown effective in some patients as a nice example of drug repurposing. But the responses vary. Not every patient benefits, and it's kind of unclear so far why some patients respond and others do not. So aside from the lipids and the coniocytes, the epidermis responds with inflammation to barrier problems. And this indicates how trained immunity, the memory of inflammation, is not a phenomenon that is restricted to the immune system, but also occurs in epidermal cells. Somehow the tissue remembers that it has seen the inflammation before. For immune cells, trained immunity enhances the ability to eliminate the infection if they see the infection again. For keratinocytes, memory of inflammation helps to restore the skin barrier. So an important function of this is when the skin barrier is breached, to recruit the immune cells. The question is how both inflammation and differentiation can be treated, how this could be treatable, druggable. Perhaps biologics have to be combined with a barrier-directed approach. I've mentioned the conified envelope when we looked at the electron microscopy images. As you know, this conified envelope consists of many differentiation proteins that are linked together by transcontaminase 1. There is a transcription factor that regulates this. AP1 coordinates the expression of the conified envelope proteins and the chemical cross-linking of these proteins. AP1 really is a complex of various proteins, FOS, FRA, various types of JUN proteins. We have shown that certain lipids that are present in the epidermis can act as agonists and activate AP1. In a gel shift assay, you can see here that certain APA components, here FRA1 and c-Jun, actually bind to the DNA response elements when exposed to a stimulus, a ligand. And this then induces keratinocyte differentiation. Ahn and colleagues have also performed these electromobility shift assays, but they did it in the context of keratinocyte inflammation. They have taken the cultured keratinocytes, exposed them to TNF-alpha and interferon gamma, and then showed that inflammation decreases the ability of AP1 to bind to DNA response elements. And they have concluded that the negative effects of inflammation on keratinocyte differentiation depends on AP1. They have found that there is an AP1 binding site in the filaggrin promoter, and the TNF interferon effects depend on it. Here, cutaneous inflammation was induced with using DNCB in mice. In the upper right, you see with DNCB a decrease in filaggrin expression, while down below there is an increase in FRA1 and c-Jun. Strikingly, the group of Richard Eckert has shown that by inhibiting AP1 transcription factor activity in the suprabasal murine epidermis, by expression of a dominant negative c-Jun through the involucrin promoter in mice, you get a scaling phenotype with changes in many differentiation proteins, but also inflammation, including chemokine increases. For example, CCL2 is up. As in human ichthyosis in this mouse model, the barrier is at risk, and the keratinocytes seem to respond by trying to repair that barrier as fast as they can. And we end up with inflammation with a stereotypic inflammatory response of the epidermis. Trying to target AP1 could modulate both differentiation and inflammation. You can directly induce differentiation, but also by diminishing inflammation, you can improve differentiation, because otherwise there is a negative crosstalk. Surely it is a challenge to find appropriate ligands. There are ligands such as liver X receptor agonists that seem to unite both pro-differentiating as well as anti-inflammatory properties that could be beneficial for the treatment of the skin barrier. So far, the anti-inflammatory effects of available LXR ligands are relatively weak, so that this is rather useful in a preventive setting, for example, between flares rather than during flares of barrier disruption-induced skin inflammation. So summarizing what we can learn by studying the skin barrier, we can modulate barrier formation pharmacologically and genetically. I've shown you studies on the role of FATP4, pointing to the possibility of specific topical fatty acid supplementation. I have talked to you about the gene editing of keratin9 mutation, which we have shown to intervene with intermediate filament clumping. And I have shown you transcription factor AP1 as a potential target for treating barrier disease. I would like to acknowledge all the people who have helped with my research, and I will be happy to discuss any questions.
Thanks a lot, Matthias. Absolutely outstanding talk, well, as expected. Please fill in the questions if you have. I'm sure you have plenty. I'm actually having one question, if I may. You know, you showed the mutation for palmoplantar keratoderma, and why is it actually limited to the hand and feet? Is this just because there's different trajectories in the differentiation of the keratinocytes? Is it that, you know, the dermis underneath is having an effect, or is I mean, the mutation should be all over the keratinocytes, but it's so clear cut? Is that known?
Yeah, it is striking, isn't it? And there was some work out of Stanford some years ago indicating that there are signals coming from the dermis. So your second possibility seems to be what's happening there, that there are signals from the dermis inducing differentially inducing different keratins, and in the palmar and plantar dermis, these signals induce keratin-9 expression.
So you think it's purely coming from the dermis? It's not, I don't know, epigenetic changes or.
I mean, I don't know how it occurs. It could well be epigenetic, and I don't know if the dermal signal is exclusive or if there's some interaction with other signals. But what we know so far is one signal from the.
Thanks a lot. Eli?
Yes. So first of all, wonderful talk, Matthias. Really, so much enjoy. So my question is actually related to FATP4 related phenotype. So and in general also, I think the same question may also apply to keratin disorders. So very often we will see a kind of evolution of the clinical phenotype. So for example, in ichthyosis with prematurity syndrome, it's very striking how the phenotype is can be extreme near birth, and then it improves with some keratin disorders like EB simplex. You have a marked improvement with time. In other keratin disorders, like with K9 associated keratoderma, there is almost no improvement. First of all, what is your take on these different trajectories? And secondly, could that be exploited also from a therapeutic standpoint?
Yes, so I can actually only speculate on this. So I think conditions that change over time, improve over time, supposedly there must be some compensation possible for the cells that only kicks in with times when the cells have matured after birth. Saying that, I'm actually thinking, there's so many different keratins. It's kind of hard to believe that there's no alternative keratin that can jump in, but perhaps that has to do with this regional specific expression of keratin-9, and there's just no other keratin that can compensate for this. In terms of therapeutic exploitation of this fact that some diseases improve and others do not, of course, if we would study what causes the improvement in those conditions that improve, that would be a key a target for treatment. So I think that would actually be an interesting approach. What makes the improved skin being better? Just as it can, I think it can be very insightful if you have different individuals in the same family and they have different phenotype severity, to study those individuals that have a less severe phenotype and perhaps identify the factors that make them have a less severe phenotype. I think this could be good for therapy if we can figure that out.
We have one question from the audience, from Ernest Ust. First, thanks you for your excellent presentation. And Peter Elias, your former boss, developed a topical lipid formula to reconstitute the skin barrier in atopic dermatitis, and if that has possibly implication or application for keratin disorders in general.
Yeah, so the skin barrier formulation copies the normal composition of the lipids that are most prevalent in the skin, which is cholesterol, fatty acids, and ceramides. So generally, using this could strengthen stratum corneum barrier function, but in a disease with a particular mutation, such as a keratin mutation, probably this is not potent enough to overcome the underlying strong effects of the mutations. So I don't think in a keratin-9 disease you have a chance to treat this with available skin barrier formulations. You have to be more specific, and even with the FATP4 mutations, I think you can't use this general stratum corneum lipid-like formulation. You have to supplement those lipids that are disturbed or that are missing.
Maybe following up on that, because one approach, obviously, and you mentioned it a bit, has pros and cons, would be CRISPR-Cas, and there might be approaches where you could just apply it locally or regionally. Do you think that that's coming in the near future, or too many downsides for a disease in the end, which is mostly not lethal?
I think there are two major points. One is how to get these enzymes into the skin if you want to use it topically. The skin barrier is so well designed to keep things out that it's also difficult to get big proteins like that into the skin. That's one big hurdle. And I think the other hurdle is safety. As I mentioned, you really want to make sure the nucleases don't do things in areas of the genome where you don't want any changes to occur. I think on this aspect, there is a lot of progress lately. These nucleases have become more and more sophisticated. But the aspect of getting these nucleases into the skin hasn't been solved so far.
Okay, thanks very much. We're already running a little bit over time, so we have to stop. If I'm getting the slides from Leo. So first of all, thanks again, obviously, to Matthias Schmuth for his excellent talk, and also Almiron for sponsoring, and obviously also Eli for co-chairing this excellent session. And next episode is actually in 3 weeks on October 4th, right, the week before EADV, and it's a sweet and sour, and two opponents will battle it off if artificial intelligence in dermatology is a game changer or actually existential threat. And it's going to be Roxana Denishu and Veronica Rothenberg, and episode will be sponsored by UCB. So thank you very much for joining. Thanks everyone for being here, and obviously mostly Matthias for this excellent presentation, which will be available in a few days also on YouTube. So thanks a lot. Bye-bye.